Good day, everyone. Welcome to our ACHEMA Pulse presentation. We're happy to have you here. Hope you are having a great time at the ACHEMA conference so far. Today, we'll be presenting on disruptive technologies in an ever-evolving biopharma environment. Kevin Smyth will be our speaker today, and if time allows, we will have a Q&A following, where Gus Dagnall will also be joining us. Please be sure you're on mute during the duration of the presentation. Also know that this will be recorded. Put any of your questions in the chat feature and we'll get to them if we have time, or we will respond to them after the presentation. Also be sure to visit our website at www.ipsdb.com. Also, for any inquiries or follow-up questions, or if you would like more information about what was presented today, you can reach me at the email address shown. I'd be happy to answer your questions and get back to you as soon as we can. Let's meet our speaker, Kevin Smyth. He's the Global Director of Process Technology for IPS. He is a chartered chemical engineer with over 30 years of global experience in the design of biopharmaceutical manufacturing facilities. He's based in Dublin, and he is part of the company's global strategy and technology group. He has led over 100 large molecule, biologics, vaccines, and sterile fill finish projects from master planning, feasibility studies through detailed design. He is an expert in drug product facility design and technology evaluation, including biological and high potent containment and cGMP production environment. Kevin has worked as a drug product subject matter expert on significant pharmaceutical projects across an ever-evolving regulatory compliance landscape. Kevin has extensive biopharmaceutical process design experience, including P&D development, HAZOP chair, regulatory compliance audits, equipment technology evaluation, and compliance with client local and global standards. He is an active committee member of the PDA and ISPE Ireland affiliate committees and regularly chairs and organizes technical seminars in Ireland for both organizations. He possesses excellent technical knowledge and a passion for peer development. If time allows, Gus will be joining us for the Q&A. Gus has over 25 years of experience specializing in the planning, layout, and design of pharmaceutical facilities. As Director of Process Architecture for IPS, his experience ranges from parenteral, fill finish, biotechnology, OSD, and bulk drug product to support facilities, including labs and containment facilities, amenity, utilities, and GMP warehousing. He has prepared and led multidiscipline teams in the preparation of strategic site and building master plans, concepts, feasibility, front-end and detailed design engineering, studies across many areas of the pharma and life sciences businesses. Gus is responsible for providing technical leadership, strategic and operational understanding to support individual projects and multidiscipline engineering teams. So that gives you a little introduction of our two experts today. We hope you enjoyed the presentation. And at this time, I'd like to hand it over to Kevin.
2, 1, action.
Thank you very much, Amy. Hello, everybody. My name is Kevin Smyth, Global Director of Process Technology and Innovation with IPS-Integrated Project Services, headquartered in Pennsylvania, US, with offices throughout the US, China, and Europe. My presentation today is entitled Disruptive Technologies in an Ever-Evolving Biopharma Environment. So, sources of contamination. Humans pose a significant contamination risk within manufacturing cleanrooms. Ideally, in facilities of the future, we would like to remove the human completely from the manufacturing floor. New disruptive technologies such as gloveless isolators, robotics, artificial intelligence, and laser-induced fluorescence can all help to reduce human interactions and improve contamination detection in the manufacturing process. Over the course of this talk, I will be presenting the latest developments in some of these technologies. I hope that you will find the material presented interesting and useful. So first, some definitions. Invention: a type of musical composition, a falsehood, a discovery, or any product of the imagination. Innovation: something new or a change made to an existing product, idea, or field. Now, new words. A disruptive technology is one that displaces an established technology and shakes up the industry, or a groundbreaking product that creates a completely new industry. Disruptive innovation, that's another one. It's the introduction of a product or service into an established industry that performs better and generally at lower cost than existing offerings, therefore displacing the market leaders in that particular market space and transforming the industry. Disruptive technology examples from our everyday life include social networking, smartphones, 3D printing, and drone technology. Uber, on the other hand, could be considered a disruptive innovation to the taxi industry, or cab industry, as you call them in the States. Disruptive technologies and innovations in biopharma that I will be presenting today include artificial intelligence, robotics, augmented reality, continuous lyophilization, and intelligent laboratories. There are, of course, many other examples, such as ATMPs and Pharma 4.0. Disruptive technologies have an important role to play in addressing market trends and requirements. These trends out there in our biopharma industry include small batches of very high value products, an increasing number of biopharma facilities producing and filling highly potent or biologically hazardous APIs, sweating the asset. That's really important nowadays. For example, multi-purpose production rooms, or increasing the utilization of high-cost manufacturing real estate. There are many, many challenges and new competing design considerations facing the industry at the moment. So what challenges do we have in the biopharma industry? Some are listed here. The big one at the moment is the impact and challenges of COVID-19. Drug shortages, both in the developed world and the developing worlds. There can be many contributory factors for drug shortages. These include manufacturing issues with impurities, particulates, sterility, delays, capacity issues, for example, facility remediation or production constraints, business decisions such as permanent discontinuation of making an individual product, natural disasters. Regarding, this has been, Annex 1 draft has been in circulation now for over 7 years. It's been through a number of consultation periods. The first consultation period, you had 6,000 comments were received. The current consultation, you had 2,000 comments received. And we think, or we believe, or we hope that it's going to be issued sometime this year, 2021. The pandemic and Brexit didn't help the issue either. Regarding cyber threats, there's many out there. For example, a cyber attack on a major filling line manufacturer in Germany in September 2020 resulted in major disruption to their business, including an email blackout for several weeks. It's estimated this year that cybercrime will cost $6 trillion, okay, with healthcare being the top target. And just as I'm going to press here, I mean, the HSE over in Ireland had a recent cyber attack which wiped out the health service for over a week. Let's discuss inspection. Talking about Annex 1, there's 2 points you'll see on the screen here. 8.29 of the current draft of Annex 1, all filled containers of parenteral products should be inspected individually for contamination or other defects. That is a requirement. You can't get away with that. The good news is, where automatic inspection methods of inspection are used, the process should be validated to detect known defects which may impact the product quality, safety, or efficacy, and be equal to or better than manual inspection methods. The gold standard is manual inspection. You can't beat the human, but technology now is developing at such a scale that AI and in particular deep learning tools are really coming to the fore. So, the future of inspection. Historically, vials, syringes, cartridges, and ampoules have been inspected post-fill, after filling, by manual inspection, semi-automatic inspection, and automatic inspection, all which continue to be used now and will continue to be used for a very, very long time. Artificial intelligence deep learning is now being applied in the biopharma industry for the inspection of vials, syringes, cartridges, etc. When compared to conventional automatic inspection machines, artificial intelligence has been demonstrated to significantly reduce the incidence of false rejects and significantly improve defects detection. Artificial intelligence is completely different from conventional automatic inspection. It involves the application of deep learning algorithms to the machine code. The good news is that artificial intelligence can be retrofitted to some existing automation machines. So, what is artificial intelligence and deep learning? There's a definition here on the screen. AI involves machines that can perform tasks that are characteristic of human intelligence. Machine learning is a subset of AI, which uses statistical methods to execute tasks without being explicitly programmed. Deep learning is a subset again of machine learning, inspired by the structure and function of the brain, which uses artificial neural networks to mimic the biological structure of the brain, capable to find patterns in data to infer the outcome of something it has never been exposed before. So the machine learns. This is like something out of sci-fi. Deep learning, by the way, is already in use by Google and Tesla self-driving cars. Facebook use it to recognize your potential friends at different scenarios. The next few slides and videos have been kindly given to me by Syntegon and Syntegon to provide you with some introductory insight into inspection using artificial intelligence. First up is Syntegon.
The human brain is something quite amazing. In complex processes, it can calibrate and manage data and reach decisions at high speed. Thanks to deep learning, artificial intelligence with a neural network is also able to conduct complex processes in the meantime. It has a number of advantages. It does not tire, is never distracted, does not require any vacation, and does not want to go home on an evening. And if it is fed the right nutrients, it passes every validation test, with significantly better results than our brain. Bosch Packaging has been feeding and teaching its latest generation of inspection lines. The inspection module is fitted with a small GPU graphic chip. This can analyze thousands of images every second. Bosch PA feeds the AI brain with some 3,000 images. The neural network analyzes the images and continues learning until the right level of knowledge has been attained. This is then frozen so that precisely the same validation result is always attained and implemented in the line. The result is very impressive indeed. In pilot lines, it has been possible to increase the defect detection to over 99%. The false rejects have been halved. And this with difficult to inspect murky fluids with a high market value and complex containers. At the same time, it is possible to create new formulations significantly more quickly because, thanks to deep learning, there is no need for the protracted manual adjustment of the inspection lines. Bosch Packaging is already piloting a dozen lines in Japan, the USA, and other countries for the use of deep learning. The same methods as those deployed to date are used for the validation.
Next up is Syntegon. October with a demonstration of their EjectX deep learning artificial inspection offering. In the next few slides, you'll see how the machine deep learning algorithm is trained to identify rejects, tested, and subsequently qualified. There is no audio with these slides, so please follow the animation. What you're seeing here is an expert inspects the training set for a specified defect. These vials are then tested on the machine to test the algorithm. Robots. Robots can do everything mechanical that a human can. They can repeat tasks consistently, are easy to be changed, are proven technology, low maintenance, and rarely, if ever, break down. They're also suitable for use in grade A environments. Challenge is the line speed with filling lines. Collaborative robots are even more advanced. They do not require barriers and can work alongside operators, although this may slow them down. The next video from SCAN shows a typical robotic installation within a grade A sterile isolator. Impressive stuff from SCAN. The next video shows augmented reality in conjunction with a packaging line. The Lion system can integrate control systems of a complete line, irrespective of what vendors supplied the individual machines on the line. Batch reports, maintenance, operations are all accessed from the same platform. else is new? Syntegon, formerly Bosch, are developing a microbatch system in conjunction with Vetter, the pharmaceutical company. We are seeing more and more collaborations like this in the industry. The long-term vision of Syntegon and Vetter is the replacement of the human with automation. This is a gloveless, suitable for smaller batches for different types of ready-to-use containers, such as vials, syringes, cartridges. It promotes loss-free filling, very important for very high-value products, fast batch turnaround with built-in intuitive operation. Continuous lyophilization, another game changer, is worth a mention, whereby the product is lyophilized or freeze-dried in advance of the filling. The resultant lyospheres can be filled into vials, syringes, inhaler powder using powder filling technology. ACHEMA have extensive conventional lyophilization experience out of the Tonawanda facility in the US, have developed this continuous spray dryer. Picture of the lyospheres is shown now. Real-time microbial monitoring. This again could be potentially a game changer. Annex 1 draft, the revised one, says the adoption of suitable rapid or automated monitoring systems should be considered by manufacturers in order to expedite the detection of microbial contamination issues and to reduce the risk to the product and therefore the risk to the patient. These rapid and automated microbial monitoring methods may be adopted after validation has demonstrated their equivalency or superiority to the established technology. And that's where we are in the industry at the moment. That's the roadblock, and that's what we're trying to work our way through. What you see here is the BioTrack system. One less intervention replacement of plates during a long fill being a significant advantage. Alternative refrigerants, again, another thing that's happening in the industry. We can no longer use the conventional refrigerants, and we're looking at alternative technologies. Nitrogen cooling has been around for a long time as well, but many, if not all, the major lyo fabricators, such as GEA, Hoff, ACHEMA, are currently developing more environmentally friendly refrigeration systems as the replacements for systems that have historically been used. But their high global warming potential refrigerants, are not really allowed anymore. These new designs are based on the Brayton cycle and can use carbon dioxide or air instead of the high global warming potential refrigerants. That's just a picture of the GEA concept. Single-use technology. I think single-use technology is well advanced now, well accepted. One of the what we're seeing now is the single-use bioreactors. They're getting bigger and bigger and bigger. Recently launched was the 6,000 liter single-use bioreactor from ABEC. Intelligent labs. Now, this is a particular interest to myself. One of my colleagues in IPS has developed this system, and we launched the Intelligent Lab at the ISPE 2020 virtual annual meeting and expo. We historically have found that lab designs are not always fully developed during design, resulting in undersized or oversized facilities. Not a lot of thought goes into lab designs really, when the focus is more been on the aseptic core. But using modern, lean and flexible lab design principles together with digital platforms, IPS, have developed an intelligent lab dashboard. If you'd like more details of our intelligent lab concept, please do not hesitate to contact anyone in our Bluebell office in the US. So, in conclusion, technology is always changing. Is change always good though? Is it too complicated? Should we look back in time on why things have evolved to the way they are and challenge the need for change? Who is driving these new technologies? Is it profit, operational excellence, or patient safety? Gloveless isolators are a fantastic idea, but we have a while to go before they're available for large-scale operation. Manufacturing capacity. Are we sweating the assets we already have? Are today's patients, our customers, being served by yesterday's technology? People are living longer and now require a cocktail of medicines. Drug shortages are a significant problem, even in the developed world, such as Germany. It is up to all of us to embrace change and bring the buzz back into pharma. We are desperately short of scientists and engineers globally. Pharma needs more top-tier scientists and engineers. We must all encourage innovative thinking, challenge the norm, embrace change, and again, bring that buzz and excitement back into pharma and the scientific world. Thank you.
End recording.
I'd like to thank you once again for joining us for our presentation today, and I hope you have a wonderful time at the ACHEMA conference for the remainder of the conference. Again, you can always contact us at www.ipsdb.com or find us on the dashboard and leave any follow-up comments or questions there. Again, thank you for joining us for our presentation.